Hey, how we doing, gang? Welcome back to Distant Learning 2.0 with Mr. Montali. This is take 749 on part 2 of our two-part series on meiosis. Meiosis, we know, is a cell division process that creates reproductive cells. In part 1, we talked about some basic terminology. We talked about crossing over, the process that creates tremendous amounts of genetic diversity, and we did an overall review of chromosomes in general. In part 2, we're going to do a complete drawing of a spermatocyte that will eventually divide into 4 mature sperm cells. Let's get to it. Now, you'll see here that we are dealing with a yellow fever mosquito spermatocyte, diploid number 6, not a human. A human spermatocyte would have 46 chromosomes that we would eventually whittle down to 4 cells having 23. In this case, with a yellow fever mosquito, we're going to take this spermatocyte and whittle it down to 4 cells that each have the haploid number of 3. Now, as you can see, we're starting in prophase 1. Interphase has happened, the cell's grown, its DNA has been replicated, it's ready to go. We're kind of in the middle of prophase 1 here. The genetic material is condensed into these nice chromosomes, and now it's time to get them paired up with their partners in a process called synapsis. So synapsis will create our homologous chromosome pairs, aka our tetrads. Let's go ahead and do that. We're going to get these guys paired up nice and neat. And get our third pair. So what we've done here is we have created 3 homologous chromosome pairs, or tetrads. Now I'm going to number them up so we can kind of follow their pathway through the entire process. We have homologous chromosome pair 1, 2, and 3. Prophase 1 is now done. We're going to move on to metaphase 1. And we know in metaphase we are going to be aligning something. In this case, we're going to be aligning homologous chromosome pairs, or tetrads. Okay, real simple. We're going to take chromosome pair number 1. We're going to take chromosome pair number, oh, that's not going to work. Oh my gosh. Number 2, and we're going to take chromosome pair number 3 and align them at the center of the cell. Nice. So they're all set and ready to go. Now, you know, of course, that chromosome pair number 1, they're homologous. They're alike in the fact that they code for the same basic characteristics, but they're different because they came from different parents. And the same for 2 and 3. So if we take homologous chromosome pair number 1 and we split them up, we know that each of those 2 cells in anaphase are going to be getting the same number of alleles and the same characteristics. Okay, so let's go ahead and do that. In anaphase 1, we know we're going to separate some things. We're going to start dividing the spermatocyte into 2 cells, and we're also going to separate up our homologous chromosome pairs. So watch what I'm going to do. I'm going to take number 1 here. I'm going to take number 2 here. And I'm going to take number 3 here. And I'm going to do the same thing on this side. I'm going to take number 1. I'm going to take number 2. And I'm going to take number, oh my gosh, I'm going to take number 3. And essentially, we're on our way to having 2 cells that have 3 chromosomes each. Telophase 1, there's some other things going on, but we're going to represent here that literally we are taking these guys, and now we have 2 completely different separate cells. And that's the end of meiosis 1. And if you look at where we started here with 1 cell that had 6 chromosomes, and at the end of meiosis 1, we now have 2 cells that each have 3 chromosomes. So that's known as reduction division. Okay, we've reduced the chromosome number in half. If this was a human cell, we would have started with 46 chromosomes at prophase 1, and then at telophase 1, we have 2 cells that each have 23 chromosomes. Okay, let's move on. So basically, telophase 1 and prophase 2 look really similar. So we're just going to take these guys and we're going to slide them on down here. Okay, so we're set there. Now, metaphase 2, once again, we're going to align something. Well, in this case, our homologous chromosome pairs have been split up. Now we have chromatid pairs, sometimes called sister chromatids. And we're going to line them up 1, 2, and 3. I didn't want to steal 1, 2, and 3. And we're also going to take 1, 2, and 3. So we got our chromatid pairs aligned, and we're going to split those up now in anaphase 2. So metaphase, we're always aligning. Anaphase, we're always separating. So in this case, we're going to separate our sister chromatids or our chromatid pairs. So we're going to do something like this. So in each of these cells, we should end up with a 1 here and a 1 there. Okay. So if we number these up and we look, we have a 1. Oops. Try that again. We have a 1 and a 1. We have a 2 and a 2. We have a 3 and a 3. Same thing here. We have a 1 and a 1. We have a 2 and a 2. We have a 3 and a 3. So each cell has gotten all of the alleles it needs. It has one full set of chromosomes. And in telophase 2, essentially, we end up with something like this. And this. And if we number these up, we're going to see we got a 1, we got a 2, and a 3. And we got a 1, and a 2, and a 3. And we got a 1, and a 2, and a 3. And we got a 1, a 2, and a 3. So each of those reproductive cells ended up with 3 chromosomes. They all have the same alleles. Okay, same characteristics, but obviously they're going to be a little bit different. Now, you might notice here that these 2 cells and these 2 cells are identical to one another, and that's true. And because I didn't show crossing over in prophase 1, that's what happens. Fortunately, crossing over always happens. And what I'm going to ask you to do is when you do your drawing, make sure you do a little bit of crossing over in prophase 1 so you end up with 4 cells that are genetically identical as opposed to what I did, having 2 cells identical there and 2 cells identical here. Okay, I hope this helped out a little bit. I'm sure there are some mistakes made. In fact, I can see one right here when I took this cell and I didn't make a duplicate of it. There we go. But it kind of gives you a general gist of what's happening. Okay. When you do your assignment, that's going to have you take a spermatocyte or oocyte with a certain number of chromosomes, take it through this process, showing crossing over. Take your time. It's a lot easier to do it when you're just doing it on paper, not having to, to put it as a lecture here. But again, this was done in one fell swoop, and hope you guys are hanging in there. Appreciate it, and we'll see you next time.